I'm Jaclyn Haven. I'm one of the genetic counselors here at Shodair Hospital in Helena, Montana, and I've been here 6 years.
I'm Dr. Elias. I'm the medical director of the Montana genetics program here at Shodair Children's Hospital. Our program is an outreach program that serves the entire state of Montana.
How do you approach a child with a developmental delay when they come to your clinic?
Yeah, so the way how we approach children with developmental delay from a genetic perspective today has changed quite dramatically over the last 10 years, 10, 15 years. In the past, we had only very limited tools available. First of all, it was very important to do a good clinical examination. So what we often did is, one of the questions that the clinical geneticists had to ask was, do I recognize a genetic syndrome? And most of the times we didn't. And then we had chromosome studies. So we were able fairly early on to look at chromosomes under the microscope. We also call this karyotype. So we all have 46, so this would be here. As you can see, we all have 46 chromosomes, 23 pairs. Girls have girls have 2 X chromosomes, and boys have an X and a Y chromosomes, and then we have what we call 22 pairs of autosomes. So those are chromosomes that look the same. They are from mom and from dad, one of each. And what we were able to look at in chromosome studies, where asking the question, is there chromosomes? Are there chromosomes that are extra? So extra chromosomes. A good example, for example, is trisomy 21 or Down syndrome. Or we could see very large pieces of chromosomes missing or extra or rearranged. The problem with that is that you see only changes that are quite significant. So, for example, an entire chromosome extra, for example, an entire extra chromosome 21, which would be trisomy 21. This is a diagnosis that we already can make just by clinical examination. When we look at the child, we can clinically diagnose Down syndrome. We don't need necessarily chromosomes to diagnose this. So we could only see very big changes, and those big changes in the chromosomes have significant consequences for the child. Usually these are children that have severe delays. They have often multiple congenital anomalies, or that's a word to express that the child has multiple birth defects. So these are severe. We know these are that there are genetic defects or genetic changes, and some of them we can see in chromosome studies. The problem with that, if you use only chromosomes, you can only diagnose perhaps 1 to 3% of children who have developmental delays. Most of our children that we see in the clinic have much milder forms of delays. They struggle with motor development. They may be late walking. They may be late speaking. They may have some mild unusual physical characteristics, for example. They may have seizures. They may have others, but they may not. Most of the children don't have other severe birth anomalies or congenital defects, although some have. So what has developed over the last really 10, 15 years is a technology that's called chromosome microarray or cytogenetic microarray. And that's a different way, simply a different way of looking at those chromosomes. If you remember the picture of those 46 chromosomes, this was a picture taken by a microscope. But another way to look at the chromosomes is to look with them with molecular technology. And what that molecular technology is able to do is it can detect the presence or absence of very small pieces of DNA. So we can actually detect small pieces of DNA or genetic information that is missing. We call these deletions. Or we can detect very small pieces of DNA that are extra. We call these duplications. And because these changes are often not visible in the microscope, we call these submicroscopic deletions or submicroscopic submicroscopic duplications. I often compare this with being in an airplane. When you think about you being in an airplane and you're kind of high above flying high, you might see kind of the coastal line and the ocean. So you might just see the ocean, the coastal line, and you see that there's a continent starting. And that would be might be what you see when you look at chromosomes. Now, when you go down further, while you're landing, for example, you start seeing individual houses. You see the houses, you see streets, and that's really what you see when you have a microarray. So you see much more detail. But when you see this, you don't see yet the people. You don't see somebody out in their yard, having barbecues, for example. So you don't see the individual people. So you don't see all of the details. So for that, you need additional technologies like gene sequencing. And we can talk about that in a different question. So what we recognized over the years is that children with developmental delay or other neurobehavioral problems, for example, autism spectrum disorder or birth defects, when you put all these cases, all these children together, and you look at these children with microarray, using microarray, you find these small pieces of DNA missing or extra, so the submicroscopic micro deletion or duplication in about 10 to maximally 20%, so between 10 and 15%, which means that if we have any child with autism spectrum or developmental delay, the overall likelihood that we find a change, what we call a copy number change. Copy number change meaning that if you have a DNA missing, this would be a deletion, this would be a copy number change, it would be a copy number loss, or if you have a duplication, it would be a copy number gain. So we find these gains and losses in about 10 to 15, perhaps 20%. The more severe the child is affected, so the more severe the delay is, the more additional features there are, epilepsy, unusual physical features, other organ involvement, the more likely is that we find such a change. So in a child who has perhaps only autism spectrum disorder but has no other features, the likelihood that we find something on microarray is likely lower than 10%. But still, overall, what we call diagnostic yield, so the likelihood that we find a diagnosis is high enough to justify the microarray as a first-tier screening tool that actually provides both screening, but also when you find something, a molecular diagnosis, and oftentimes and also a clinical diagnosis, utilizing the microarray.
An analogy I like to use with families when we talk about chromosomes and the different testing options is you can think of the chromosomes like books, like a set of encyclopedias in every cell of the body. And so the karyotype, like Dr. Elias talked about, was looking to see, is every book there in the set? Are there any extra books or missing books? And that was really all we could see. The chromosomal microarray is looking in the book to say, essentially, are there any pages that are missing or extra? And so it's a much deeper detail, but we're still not looking at the words. And when we do look at the words, to look at, are there any missing or extra words, any extra missing letters, that's really when we're talking about single gene sequencing and multi-gene panels, as well as whole genome sequencing.
Thank you so much for sharing the insight about chromosomal microarray as a first-tier test for a child with a developmental delay. What are some of the other things that can be done for a child if that step does not yield answers?
Yeah, so this is a good question. I mentioned earlier that with the chromosome microarray, we find across the board, so when you look at all of children with various degrees of developmental delay, autism spectrum disorder, or maybe birth defects, you find changes clinically significant changes. So basically, we found a diagnosis in between 10 to 15%, perhaps 20% in selected groups, which means that, you know, in at least 80% of children with developmental delay, we do not arrive to a diagnosis with chromosome microarray. So what do you do in that case? So it depends. It depends really on how the child presents, and this is then where we have to put our clinical hat on and see how does the child present. So we have to ask again, are there features in the child who that lead us to a certain diagnosis? For example, there is children with delays. Some children with delays have certain physical features and also certain heart defects that lead us to think about a diagnosis of Noonan syndrome. So Noonan syndrome is a recognizable genetic syndrome. It's caused by a limited number of changes in a limited number of genes. When we think about Noonan syndrome and there's enough clinical suspicion, what we then would do is we would look at those genes that when you have a change in those genes, you might have Noonan syndrome. So this is a gene panel. Jaclyn earlier mentioned the idea of a book, a page, and perhaps a recipe. So we quickly have to talk about what a gene is. A gene is basically a recipe to make a protein. A protein make up our body. So when you can imagine if there's a change in that recipe, a mistake in that recipe, the protein might not be done in the right way. And if this is in a protein that is important for Noonan syndrome, then you might actually have Noonan syndrome. So what you would do then is you would look at changes. You would read the recipe to find changes in the recipes of these genes that are associated with Noonan syndrome. So this would be a limited gene panel. And it's important to note that if we do a good clinical exam, then we often, not always, but frequently can determine or can suspect a number of different syndromes that then let us do a number of different gene panels or a number of different genes that together make up a group of genes that we would call a gene panel. So this would be a limited gene panel. The advantage of a limited gene panel is that it's quick, it is inexpensive, and it gives us, it is mostly focused on the answer, meaning that we do not get answers that we don't want to know about, meaning that the likelihood of what we call a variant of uncertain significance is lower. So if we often, and I would say probably most of the time, when we have a child with developmental delay for whom a microarray is negative, the child presents quite nonspecific, meaning that there is significant delay. There might be seizures, but there are no specific physical features that would lead us to a certain syndrome. And one thing that we have recognized over, again, the last 5 to 10 years is there's between the different genetic syndromes, there is a lot of overlap. That means that you can have a change in different type of genes, but children present very similarly, which makes it very difficult to actually look at, to actually make a clinical diagnosis. And in this case, what we call a genomic approach to sequencing, so a genomic approach to inquire all of the recipes, all of the gene that make up our body might be the best approach. And this is what's called whole exome sequencing. So whole exome sequencing, you might have heard, is basically looking, to use Jaclyn's analogy of books, recipe books and pages, looking for errors in the most important chapters in the book. So what does that mean?
First, our human genome contains approximately 20,000 genes. So every cell in our body has the information of these 20,000 genes. And if you remember, in general, each gene carries the information or the recipe to make a protein. We say a gene encodes a protein. However, only parts of each gene actually contains the information to make a protein. Those protein coding parts of each gene are called exons. Introns are also part of the gene, but they do not carry information to make protein. So typically each gene has several exons separated by introns. Second, if you combine the information of all exons together, we call this an exome. It's worth noting, though, that the exome, so the combined information of all exons of all 20,000 genes, only represents about 1 to 2% of the entire human genome. In other words, only 1 to 2% of the entire human genome is protein coding. And then third, we know from experience that most disease-causing genetic changes are in exons, so in the protein coding portions of the genes. So focusing only on the 1 to 2% of our entire genome, the exome, at least for the last 10 years, has been the most efficient way to interrogate or to test our genome for disease-causing genetic changes. But it's worth keeping in mind that whole exome sequencing still only looks at the information of a tiny part of our genome.
And that's why we're talking about whole exome sequencing when we attempt to sequence, to read the information of all of the protein coding parts of our 20,000 genes. Now, when we do this, when we have a child with developmental delay, non-specific developmental delay, and we apply whole exome sequencing to it, depending on the situation, depending on the age of the child, depending on the type of presentation, how severely the child is impacted, and in part depending on the platform, depending on the technology, we might find a diagnosis between 20 and 60% of cases. So with microarray, we might find a diagnosis in between 10, 15, perhaps 20%. And with whole exome sequencing, we find it between 20 and 60%. So this is a significant increase in the diagnostic, what we call the diagnostic yield. What it also means is that we still have 40% at least where we don't have a diagnosis. And among those, we don't know where the change is, but likely in some of those, in a subset of those 40% where we don't have a diagnosis with whole exome sequencing, this will be the change we might find in those non-coding parts of genes. So those genes, those parts of the gene that don't end up in the actual recipe. And then also in those parts that are between genes. So only about 1 to 2% of our entire genome is actually protein coding. So 98 to 99% of our genome is non-protein coding. When we assume that in those 40, at least in a subset of those 40% of children where we cannot make a diagnosis using whole exome sequencing, the change might be found in those 98% of the genome, the non-coding genome basically, that we don't capture with whole exome sequencing. That is captured with whole genome sequencing. With whole genome sequencing, we literally attempt to sequence the entire genome. So we're looking basically for errors, just to use Jaclyn's analogy again, we're looking for errors in every single word in the book. That again increases the diagnostic yield. Whole genome sequencing is increasingly used in genomic centers. It is still mostly used either in the research setting or for certain applications. It's not used in a routine in a routine fashion quite yet, but this likely will change.